Greetings and welcome once again to another session. Today we're going to be continuing our conversation in relation to the ABO system. In the last discourse, we established that the ABO blood group system is the most important of all the blood group system. And we established that it was important in both transfusion and transplant medicine, all right? And we also disclosed that it is the only blood group system in which individuals already have antibodies in their serums to antigen that are absent from their red blood cell without prior exposure to red blood cell through transfusion or pregnancy. So this basically means that this blood group system, majority of the population is born with it, all right? And if you don't have it, then you are placed in another category, which is the lookalike to the ABO, which we classify as the Bombay. Due to the presence of these antibodies, transfusion incompatibilities can be derived. So if someone who is A gets B, that's a problem. If someone who is B gets A, that's still a problem. If someone who is O gets A, gets B, gets AB, that there is a problem. If someone who is of a blood group that they are receiving from someone else who is not of the same blood group, that can cause some conflict, and there is going to be antigen antibody complex formation because the body is going to try to protect itself, and in protecting itself we're going to see a situation where the body's defense system goes on attack, and when it goes on attack it creates these clump which we call agglutination, and these things may result in blood clots form. But what happened at the end of it all is that it causes the organism to come to its demise. So we have to be very careful and mindful in ensuring that the blood that is transfused is one that is viable, one that is suitable for the purpose intended. So ABO blood group system is a system that you want to be most proficient in, because this ABO testing is done in almost all blood banks. All right, this is the first step in determining a person's viability for transfusion. All right. So even today, transfusions can go wrong if there is an ABO incompatibility. All right. So we have to just bear this in mind. So we have Carl Landster to thank for the ABO blood group system, at least based on documented evidence. It was in 1901 that Landster drew blood from himself and 5 associates. He separated the cells and the serum and then mixed each cell sample with each serum. So when we're thinking about him mixing the cells and the serum, we're thinking about the forward and the reverse blood grouping. The forward blood grouping is the front grouping. Is defined as using a known source of commercial antiserum, which is the anti-A and the anti-B, all right, to detect antigens on the individual's red blood cell. Because remember, the red blood cells, let's bring it up. The red blood cell, we're going to put it there. This is the red blood cell that we're looking at. On the surface of the red blood cell, these are the antigen binding site that we have right here. These are the antigen binding site, and each of them have their own specific look. All right, and that is why we have the goodness to fit, because specific antigens bind with specific antibodies. All right, very good. So now the reverse blood grouping or the back typing is what we considered to be the method of detecting ABO antibodies in the patient's serum by using reagent red blood cell. All right, so we are doing the opposite. And basically, we normally use the A1 cells and the B cell, but there are different subgroups of A's, and depend on where your testing is done, depend on what the predominant blood grouping is, then you may have to use the subgroups like the A2 or the A3. All right, so it all varies, but the idea is that you're going to have A cells and B cells in order to determine what anti antibodies are present. All right, so the A cells and the B cells give us an idea so that we can compare it with the forward group, because remember what we established first. If they have A antigens, then they will not have A antibodies, because if they have A and A, it's going to come together and it's going to form agglutination, and then that will lead to the termination of that individual's existence. So if the person has A, we expect them to have A antigens, we expect them to have B antibodies in circulation. If the person has B antigens, we expect them to have A antibodies in circulation. If the person has no antigens on the cell or their own, we expect them to have both A and B in circulation. If the person have both A and B antigens on the surface of the cell, then we expect them in circulation to have no antibodies. All right, so that is just an idea as to how things work. So let us go back to it. This is a cell, and on the cells we have the antigen binding site, and each antigen has its own unique look. So the A people have a specific look for the A, the B have the specific look for the B, and that is just how it is, right? So we have established the forward blood grouping, and we have established the reverse blood grouping. So these two blood grouping are still a part of the ABO blood grouping system, and that is just one blood grouping system. There are other blood grouping systems that are implicated in transfusion reaction, but they are not necessarily at the room temperature in which they are identified. They have to be taken through some other testing methodologies to determine compatibility. And we are going to go into those in a little while, but we just want to focus on the ABO blood group system. So ABO blood group is the most frequently performed testing in the blood bank. It's the most frequently performed testing. Both the ABO forward and reverse grouping testing must be performed on all donors and patients. So the donor has to be tested, the patient has to be tested, because this is the simplest form of compatibility. We want A people to receive A blood, B people to receive B blood, O people to receive O blood, AB people to receive AB blood as best as possible. But there are instances where we can give them different blood based on what is available and what is compatible. All right, because you know that let us go back on our board. An O person can receive blood from an O individual, right? A B person can receive blood from a B person or an O person. That is the packed cell that we're talking about, because remember, the O person has no A or B antigens on their cells, So they can give the red cells to a person who is B. A person who is A can also receive blood from a person who is A and O. That is correct. So we're doing very good. Now, this is where the caveat comes in. A person who is A and B can receive from a person who is A, can receive from a person who is B, and can receive from a person who is O. And you're saying, wow, this there then puts this individual as the universal recipient. So the AB person can receive blood from any of the blood groups. The O person, we realize that O is right here, O is right here, O is right here, O is right here. So this O person can be considered to be the universal donor. So the O person is the universal donor, which means that they can give to anybody. And the AB person is the universal recipient, which means they can receive blood, the packed cell portion of the blood, from anyone. All right? That's all we need to bear in mind in this moment in time. Now, as we move further along into the ABO blood grouping system, it has been postulated that in some instances bacteria, pollen particles, and other substances present in nature are sometimes chemically similar to the A and the B antigens, right? So bacteria are widespread in the environment, which constantly can create, in some instances, some exposure and present some A-like observation or B-like antigens being observed. This exposure serves as a source of what we call ABO discrepancy sometimes, because a person who is A should have a particular blood group in the reverse observation. A person who is B supposed to have a particular in the reverse observation. But sometimes environmental factors can cause someone who is O to look like someone who is A. So then there is some incompatibilities with the forward and the reverse, which would lead to further investigation to determine what is the cause of this situation. All right, so we just have to bear that in mind. All other defined blood group system do not regularly have naturally occurring situation taking place. They are usually based on exposure to something, right? And the body then produce the antibodies based on the reaction or the interaction with the antigens. All right, so just bear that in mind as we move further. Let us talk about the ABO antibodies. Individuals normally produce antibodies directed against the A and or the B antigen present on their red blood cell. So basically, as we established before, if you are A, you're gonna have the opposite, which would be B antibodies in circulation. If you have the A antigens, you have the B antibodies. If you have the B antigens on the cells, you're gonna have the A antibodies in circulation. So this is just the natural way how we have been designed to preserve our existence. So let us just bear that in mind. So let us move forward now and understand that the ABO antibodies are predominantly IgM. Yes, and they are activated by complement and they react at room temperature and can even react in cold environments, right? So ABO antibodies produce strong direct agglutination reaction during the ABO testing. ABO antibodies produced is initiated at birth. However, though we are born with all of this, when we are under 6 months, the body has not yet mounted enough titer or enough concentration to give us a reliable and reproducible result when this testing is performed. So what we do for the babies and for the geriatric is sometimes for the babies we do not perform the reverse blood grouping if the individual is under 6 months. For the adults, geriatrics particularly, when they are over a certain age, like they are getting to 70, 80, 90, 100, when they're in that age, we tend to find that there is a decrease in the reactivity in the reverse blood grouping. And this there is usually due to the fact that as we get older, our antibody production is reduced. And because of that reduction, we may have some challenge in confirming the forward blood grouping with the reverse blood grouping. But understanding the patient history gives us an idea as to what are possible causes of the discrepancy that is being observed. So, to perform the ABO blood grouping system, we need the anti-A reagents, we need the anti-B reagents, we need the A cell, usually the A1 cell, and we need the B cell. All right. So the A reagent is usually monoclonal, it's highly specific, it's usually IgM, and the color that is used to denote the A is a blue coloration. The expected reaction is usually 3 to 4, and we usually do 1 to 2 drop depending on your laboratory directions and the operation procedures which they are going by. All right. So for the anti-B, these are usually monoclonal antibodies. Yes, they are highly specific. They are IgM as well. The color though for the B reagent is usually yellow, and it contains a dye. The dye is called archiflavin dye. So that is the dye that it contains. All right. So the expected reaction still is 3 plus or 4 plus, and usually it's the same 1 to 2 drops that are used. For the reverse blood group in the A1 and the B cells, it is usually derived from human sources. So persons who are A, they are the ones who we get the A cells from. But we usually use the A1 lectin to determine persons who are A1, and we get the blood from that source and pool them together. But in some laboratories, there are commercially prepared A1 cells, which makes it easier for the laboratory to operate. So they don't have to be preparing their cells to use their cells. They can just source their cells and have them quality controlled from the preparer's laboratory while they just use and enjoy the benefits of the cells. All right. So we usually do 3 to 5% suspension. Some may say 4 to 5% suspension, but it varies based on the location, based on the concentration, based on the preparation. So the expected reaction with the A1 cells and the B cells is usually from 2 plus to 4 plus, and it's usually one drop that is utilized. All right, so let us just do a quick summary of the forward patient cells with the reagents. So let us go back on our board once more. So let's say the blood group, and we're going to say anti-sera. This is for anti-A. Then we're going to say this is anti-sera B, and we're going to come to the conclusion of the antigens on RBC, red blood cells. All right, so a person who is O, we are expecting no reaction with the A, no reaction with the B, right? Because there are no A or B on the red blood cells, so there is none. All right, a person who is now A, we're expecting a reaction in A, no reaction in B, because this person has A antigen. Now a person who is B, so we get in the picture, we're expecting no reaction with the A, we're expecting a reaction with the B, and it is because there is B antigens. Now a person who is AB, we're expecting a mixture of reactions. We're expecting a reaction with the A, and we're also we have a reaction with the B as expected, because this person has both the A and the B available. All right, so with that observation, with that understanding, we have an appreciation as to what is taking place. Now this is a forward. Let us write it forward, FORWARD. So this is a forward blood grouping. Now let's move to the reverse blood grouping. All right, so this is the reverse, REVERSE blood group. All right, so we're going to be using cells in this instance. So we have the A1 cells and we have the B cells, and this is going to give us which antibody or bodies exists in the serum, right? So the person is O. We're expecting to have a 4 plus reaction in the A cells, a 4 plus reaction in the B cell, because there are A and B antibodies in circulation. A person who is A, we're expecting no reaction with the A, but we're expecting some form of reaction with the B, because there is B in circulation. When a person is B, we're expecting a reaction in the A, but no reaction in the B because they have A. Now, when a person is AB, we're expecting no reaction in the A and no reaction in the B. So we are going to put none. All right, so when we're looking at this now, we're seeing none, none. So this here, for the AB person, this whatever liquid portion of the cell that exists, it can be given to everybody. All right? So that is just something that we need to understand. When a person is AB, they can give their plasma portion to anyone. And that is just something that we have to understand and appreciate. All right? So that is, in a nutshell, how the ABO blood group system is. But I must bring to your attention that when it comes on to the phenotyping frequency, there is different ethnicity that has predominant in certain blood groups. All right? So let us talk about the phenotype. The phenotype just basically denotes the O, the A, the B, and the AB, right? That's the phenotype, what we observe, right? So a white person or a Caucasian person, then we have a black person, we have the Hispanic, and we have the Asians. We are observing that 45% approximately of the O exists in white population. The white population, 40% A, then 11% B, 4% AB, right? For the blacks, we have 50% O, so majority of the black population is O. Then we have 26% which being A, then we have 20% being B, and we have 4% being AB, just as in the white population. So the black and whites have the same AB ratio. Now, for the Hispanics, we have more persons, 56% being O, and then we have 31% being A, we have 10% being B, and we have 3% being AB, right? Now for the Asians, we have 40% being O, we have 28% being A, 25% approximately being B, and AB is 7%. So we are realizing that the Asian population have more AB, while the one with the lowest would be, for the AB, would be the Hispanic population. In the same breath, we realize that majority of Hispanic individuals will be O, all right? But when it comes on to O blood group, we are only expecting 40% of the Asian population. So this is just something that we need to know and just have an idea. So when you go to the physician or when you go to the provider and they indicate to you that they need to know your race and ethnicity, it's not always because there is some discrimination taking place. Sometimes when it comes on to a medical procedure, it just gives an initial overview as to what can be expected. And when this information is derived, your treatment can be tailored in such a way that it is beneficial to you and it provides you the best opportunity for a fast and speedy recovery. All right? So just bear those information in mind. We could go into the offspring determination for the ABO, but we're going to leave that for just a little moment and understand that when a person has A blood group, they have a particular marker, right? When a person has B, they also has a particular marker. And when we say marker, we're talking about sugar because these are just sugars, right? When a person is O, we classify them as H. They also have a particular marker, all right? So a person who is A has this marker. A person who is B has this marker. And a person who is O or H has this marker. And as it relates to the immunologic sugar or the immunodominant sugar, a person who is H, the sugar is L-fructose. The person who is A, the sugar is N-acetyl D-galactosamine. All right? And then a person who is B, it's D-galactose, all right? Galactose. So this is just something that we need to understand, and it's because they have the different antigen. So based on which antigen they have, and then this here is the immunodominant sugar. So these are just some things that we need to understand as it relates to the ABO blood group system. So we need to go into the sugar a little bit more so that we can have a better understanding and appreciation. But we're going to take this up in our next sitting because, as you know, class is dismissed. Until we meet again, continue to study, continue to show yourself, approve, and at the end of the day, when the results have been determined, your results will be indicative of the invested time that you would have deployed into learning this material. We're going to go through some cases and some questions that are used to gear you for the examination, the ASCP examination that is a possibility for majority of those of you who are observing and understanding. It is my intention to provide a resource, but before I provide the questions, I want to provide you some guidance in theory so that you can use the information you receive to answer the said question and apply the knowledge as is necessary. Once again, thank you very much for spending some quality time with us, and do enjoy that which has been as we anticipate that which is to come. Class is dismissed.